जैसा कि हम कल्चर मीडिया के हवाले से बात कर चुके हैं, बैक्टीरियल कल्चर और माइक्रो ऑर्गेनिज्म कल्चर वंस इट इज़ एस्टेब्लिश्ड, बायोटेक्नोलॉजी में जो नेक्स्ट स्टेप है दैट इज़ टू isolate the DNA for further study. This study can be of different types, but we are at the moment going to talk about the isolation of DNA, how many steps are involved and how we can go further with isolation. As a general principle, using a bacterial cell to isolate the DNA, we have to go with 4 different steps. The first step is going to be the bacterial culture. Once you establish the culture using the defined or complex culture media, then the density of the bacteria reaches to a certain level. We have to harvest those bacteria or any microorganism. Once the microorganisms are harvested, the next step would be break down their outer coverings, the cell walls, so that the cellular components are released outside in the solution and we can further proceed. The treated cells or the ruptured cells are going to be processed further so that to remove all the components except the DNA. Once we isolate the DNA from rest of the debris, cellular debris or any cellular components, we can further concentrate it and we can obtain the purified DNA. Bacterial cell or the microorganism cells, they have 2 types of DNA, genomic DNA and the plasmid DNA. In biotechnology, most of the time when we are working, we need plasmid DNA. In biotechnology, there are different methods to prepare the plasmid DNA. This picture shows you a summary of the steps how we can start from the bacterial culture and we can end with the required DNA. The culture, centrifugation, then you can see the harvesting of the cell, which means all the cells are going to be settled down in the tube. The supernatant is discarded. The bacterial cells are processed further to destroy their cell walls. Cell extract is prepared and further processed for pure DNA separation. The plasmid DNA can be obtained by different methods which can be referred as mini preparations, midi preparations, maxi preparations, mega preparation or giga preparation. In the laboratory, the most commonly used method is a mini preparation, which is used for a small scale DNA isolation, and the expected yield comes up to 50 to 100 micrograms. As the sample size increases, means the volume of the bacterial culture which you are using is going to increase, the quantity of DNA which is going to be produced is going to increase, and we have to decide, keeping in view the volume of the bacterial culture, which method to employ, the midi, maxi, mega or giga preparation. The giga preparation can isolate the DNA from 2.5 to 5 liters of the culture media, and the expected yield comes up to 7.5 to 10 milligrams DNA per milliliter.